Good day, ladies and gentlemen, and thank you for standing by. Welcome to OS Therapies Business Update Conference Call. I'm Harrison Seidner, Director of Investor Relations at OS. In addition to myself, with us today are Paul Romness, our Chairman and CEO, Dr. Robert Pettit, our Chief Medical and Scientific Officer, Gerald Commission, our Chief Business Officer, and Dr. Sir David Brindley, our Lead Regulatory Strategy Consultant, and Dr. David Brindley, our Regulatory Strategies Physician. This time, all participants are in listen-only mode. A brief question and answer session will follow the formal presentation. If anyone should require operator assistance during this conference, please press the star followed by the 0 on your telephone keypad. As a reminder, this conference is being recorded. Before we begin, I want to remind listeners that during this call, management's prepared remarks may contain forward-looking statements and are subject to the risks and uncertainties. Management may make additional forward-looking statements in response to your questions today. Therefore, the company claims protection under the Safe Harbor and forward-looking statements contained in the Private Securities Litigation Reform Act of 1995. Actual results may differ from results discussed today, and therefore we refer you to detailed discussion of the risks and uncertainties in the company's filings with the SEC. In addition, any projections as to the company's future performance represented by management include estimates as of today, October 13th, 2025, and the company assumes no obligation to update these projections in the future as market conditions change. This webcast will be available on www.ostherapies.com in the Investor Relations page. I want to turn the call over to our Chairman and CEO, Paul Romness. Paul.
Thank you, Harrison, and welcome everybody. Thank you for joining us today. I'm Paul Romness, Chairman and CEO of OS Therapies, and I'm truly excited to share with what has been an extraordinary few weeks for our company, and most importantly for patients and families we serve. Our team has worked tirelessly, and the results from our OST-HER2 program have been inspiring. Just last week, we announced statistically significant and clinically meaningful final 2-year overall survival data in our phase 2 trial for OST-HER2 and recurrent, fully resected pulmonary metastatic osteosarcoma, an area that has not seen a new therapeutic option in over 40 years. For the first time, we believe we are seeing a genuine shift in what survivorship can look like for these patients. OST-HER2 treated patients achieved a 2-year overall survival rating of 75% compared to only 40% in the historical control group, with a p-value of less than 0.0001, a strong database that we believe represents the key clinical efficacy data around which a full database will be delivered to various regulatory agencies to support conditional marketing authorization in the UK, the United States, and the EU. We intend to pursue additional jurisdictions once we have confirmation on the regulatory path in these initial markets. We've now had multiple regulatory interactions with the FDA, MHRA in the UK, and the EMA in the EU. Each of which confirmed that overall survival is the overarching clinical endpoint around which efficacy data should be evaluated and is an acceptable clinical endpoint to support conditional approval. We're preparing for key submissions: a conditional marketing authorization to the MHRA this December, an accelerated approval BLA to the FDA in January, and our EU submission shortly thereafter. This sets us on a path toward our goal of becoming a fully commercial biotech company in 2026, with OS Therapies leading the first meaningful change in osteosarcoma care in generations. So today, as we look ahead, I want to thank our team, our investigators, our partners, our shareholders, and our patients for their steadfast belief in what OS Therapies represents: a rare company with both heart and science, steadfast in our pursuit to save lives. This is an exciting and defining moment, not just for OS Therapies, for all of those waiting for hope. I'd like to now hand it over to our Chief Business Officer, Gerald Commission. Gerald?
Thank you, Paul. We want to remind everyone that OS Therapies is developing immunotherapies for cancer based on the pioneering work of researcher Dr. Yvonne Patterson from the University of Pennsylvania. OST-HER2, an attenuated live listeria immunotherapeutic agent engineered to selectively infect HER2-presenting cancer cells, is the company's lead product candidate and is being developed for the prevention or delay of recurrent, fully resected pulmonary metastatic osteosarcoma. Within this disease setting, the company completed a phase 2b clinical trial that enrolled 41 patients, with 5 patients lost to follow-up as of the 2-year mark, with 40 patients evaluable at the 1-year time point and 36 patients evaluable at the 2-year time point. There are approximately 1,000 patients diagnosed with osteosarcoma in the United States, diagnosed primarily in adolescents. Osteosarcoma has been classified as a rare pediatric disease by FDA. The clinical path for these patients involves the resection or amputation of the limb after their initial diagnosis. And that resection is where the primary tumor is found, combined with about a 9-month chemotherapy regimen. Following chemotherapy regimen, there is an approximately 40% relapse rate, with relapses occurring primarily to the lungs, with worse outcomes seen in patients whose lung metastases are identified within the first 2 years of the resection of their primary tumor. In this patient population, approximately 400 patients annually exist in the United States, and their prospects for survival drop dramatically when they become metastatic, as the standard of care remains surgical pulmonary tumor resections, with additional metastatic recurrences typically expected within 3 to 6 months after each metastatic event, if they are to happen. While there is significant heterogeneity in this patient population, a majority of patients expire prior to reaching 2-year post-initial metastatic event in every single peer-reviewed publication our team has identified throughout the literature. As such, OST-HER2, with recent 2-year overall survival data of 75%, may represent a compelling treatment option for patients if approved. The team is working hard to put this into the proper context for regulators worldwide. After several conversations with FDA on the path forward, being able to clinically interpret this data to support a conditional marketing authorization via accelerated approval, the company is now preparing for what we believe will be final rounds of meetings in December with UK, MHRA, FDA, and EMA. And with all 3 regulatory agencies prior to submitting conditional marketing authorizations in the UK in December, the FDA in January, and later on in the first quarter of 2026 in Europe. The meetings later this year will be focused on gaining alignment that the data that we will be presenting meaningfully improves upon the standard of care, with a high degree of certainty that this is being attributed to the treatment rather than by chance. As such, we've taken extra steps of care over the weekend after some recent FDA meetings to confirm that the patients enrolled in our trial are generally representative of the metastatic osteosarcoma patient population, and we've identified no bias in those patients enrolled. We fundamentally believe that the immune system training mechanisms of action of our drug is likely to drive overall survival more meaningfully than it is to drive event-free survival, because of the persistence of immune response after antibiotic intervention. While the trial design was driven largely as a result of prior experience from metastatic osteosarcoma trials from our clinical partners, rather than based on the mechanism of action of our drug, the continued overall survival listeria monitoring follow-up has allowed us to capture data indicative of a statistically significant overall survival benefit when compared with all available databases. From a timeline standpoint, with conditional marketing authorization applications expected in the coming months, the company is preparing for commercial deployment in the United States with our contracted partner, Eversana. We remain engaged in strategic partnership discussion with other biopharmaceutical companies, and as we make progress towards accelerated and other approvals outside the US, we think we may be able to secure partnerships. We are still in a position to be granted a priority review voucher around our OST-HER2 osteosarcoma program, and that voucher has reached a market value of approximately $160 million based upon the most recent transaction earlier this year. The company is also continuing to evaluate strategic transactions for the canine OST-HER2 osteosarcoma rights, and expects that some new data to be published in the near term from Dr. Nicola Mason's lab, which we gave top-line data on in early April, will really help drive this. Further, the company's pipeline of listeria immunotherapy candidates added via the Ayala Advaxis acquisition allows us to have another important clinical endpoint coming in the prostate cancer program later on this quarter. While osteosarcoma represents a patient population of only 1,000 patients, and we're going after a subset of a little over 400, with about 10% of the patients represented in our trial. Prostate cancer is over 300,000 patients annually, with over 30,000 patients passing. And so that represents a very significant unmet medical need for a much larger patient population that our company can go after. Last week, the company participated in an individual OST-HER2 EMA rapporteur meeting on Monday, and on Friday, we were at the Osteosarcoma Institute FDA Osteosarcoma Workshop in Washington, D.C. I will turn it over to David Brindley at BioLacuna, the company's regulatory strategy advisor, to give some background on turning signal-finding studies in rare diseases into market authorizations via enabling data, with the promise of follow-up clinical studies. This is a key pillar of our regulatory strategy. Members of David's team participated, obviously, both in the EMA discussion as well as the FDA workshop, and we'll put into context some of the key points that came out of that meeting, especially with regards to randomization, the translational nature of the canine model, and the importance of overall survival to patients and regulators. A key point from last week's meeting was that all other efficacy endpoints are really just surrogates of overall survival, and given the mechanism of action of our drug, overall survival is where the focus needs to be. With that, I'll turn it over to David. David?
Thank you, Gerald. Good morning, everybody. So I'm on the phone today with a colleague, Andrew Exley. So both of us are regulatory affairs experts, and in Andrew's case, he's also a MD, PhD immunologist and ex-clinical assessor in Europe for a national competent authority. I think there's been tremendous in building of momentum across all OS therapies programs, particularly the osteosarcoma candidate. And I think that something which is very encouraging is that of the three aspects of a regulatory submission, so non-clinical, clinical, and manufacturing, non-clinical and manufacturing are very much in good shape with the regulators. And in some cases, we've actually started to commence pre-submission activities there. And from a clinical perspective, we now, in the context of the information Andrew will talk about in a second, we're looking to understand how the regulator wishes for the data to be presented, and then secondly, any post-market activities that the regulator will require. One thing which I think is important to stress here is from the perspective of the single-arm study that was done. Single-arm studies are very common in rare diseases, and if we take just Europe as an example, there were 18 approved medicinal products just for solid tumors alone on the basis of single-arm trials between 2012 and 2021. So this is a very well trodden path, albeit in a very rare disease. Andrew, could I possibly hand over to you to talk about the pillars of the regulatory approach, in particular, the safety, the animal models, and the MOA?
Thank you. Very happy to, and good morning to everybody. Can I just check that you can hear me all right, please?
Loud and clear.
That's great. Thank you very much. So the attenuated listeria recombinant immunotherapy as a platform is very well described. You may not know, but this bug is something of a favorite amongst immunologists and infectious diseases experts because so much has been learned about the mechanism of the immune response to this bug. And in fact, this platform has been used now in over 1,000 patients, although clearly the specific HER2 analog has been rather more focused. As an immunotherapy, quite often you struggle to get a good animal model. And so we're delighted to have such an excellent animal model in the form of canine osteosarcoma. which has provided a rich source of data and very much underlines our understanding of the mechanism of action of this therapy. Perhaps unusually as well, there's a rich literature on overall survival from the SEER database. Rich, but unfortunately rather depressing in some respects, in that there's literally been no improvement either in overall survival from presentation or overall survival from metastases over the last 30 odd years. And these data and this analysis really unlocks the ability to look at the literature and to use slightly older papers perhaps to inform on the likely overall survival without this sort of intervention. Added to that is a further pillar of the clinical data itself, which you've already heard outlined, not only in terms of overall survival, but the likelihood that we should be able to use an additional biomarker-based approach inspired by the canine studies to shed further light on the efficacy of this therapy. I should also refer back to the safety data based on exposure of over 1,000 patients, because one of the pieces of information that helps in judging the whether to approve or not these sorts of therapies is what can you tell us about safety? And so being able to draw on a rather larger database for the platform analysis is extremely helpful.
Yes, we could pass it to Paul for closing remarks before we do questions.
Sure. When we get back to it, Andrew, it'd be great if you could talk about biomarkers and that's interplay with overall. So thank you for all those insights. I want to take a moment to reflect on just how far we've come, not only in the past quarter, but in the momentum that builds across every aspect of OS Therapies. The dedication, teamwork, and innovation of this team fills the halls with translating directly into meaningful advances for patients and families who for too long have been left waiting for hope. As we move forward, let's keep our focus clear. We are on the precipice of achieving something transformational for our community and for rare cancers in general. The science, the execution, and the compassion that defines this team are now being recognized by regulators, clinicians, and most importantly, patients. We have a responsibility to see this progress through, to deliver on our promises, and to continue developing value for all our stakeholders. Thank you to our team, to our partners, and to our investors for believing in our mission. We are grateful for your continued support and look forward to continuing to share updates with the market. Harrison, this concludes our remarks. If we could open it up for questions, that'd be great.
All right. I'm going to open the floor for questions right now. If anybody has any.
Yeah, I think let's take a question from Chad, Jan, as it related to the biomarker. Do you see that question, Andrew? I think you can go into that. I'll read the question aloud. We were wondering if you could go into more detail on the biomarker side.
Thank you very much. Yes, the analysis from the canine osteosarcoma study really underpins what we expected to find based on the mechanism of action. And that is that the responders to the immune therapy show a very strong upregulation of the appropriate cytotoxic T cell genes, NK cell and gamma delta cell genes, so that the observed clinical effect seems to match really quite closely what you'd expect in terms of the immune response that's been stimulated by the therapy.
Thank you. Robert, I know you've got some background and experience with the biomarker side, if you'd like to add a little bit to that.
Yeah, absolutely. I think Andrew, of course, has hit the nail on the head. The whole objective of this type of therapy is to generate a potent stimulation of the arm of the immune system, or arms of the immune system that are responsible for tumor surveillance in a targeted way. In this case, starting with HER2, and then as cancer cells are destroyed by other T cells in this environment, then additional tumor antigens are picked up, and the idea is that this constant the immune system is there battling against the progression of the cancer, and this sort of war, if you can imagine it going on inside the patient, slows the ultimate growth and sometimes stops the growth of the cancer and allows that patient to survive long term. Early on, it may or may not be given in time to stop the next metastasis that's on the way, but subsequently it just keeps building up that immunologic pressure against the progression of the cancer. And the patients who do the best on this therapy are the ones, not surprisingly, who have the more robust immune response to the therapy itself, because that's really the whole concept behind immunotherapy. Let's turbocharge your immune system to make it more capable of fighting against this cancer, and in the process extend life and quality of life for patients. And so here we have something that appears to be doing just as it's intended to do. And the biomarkers were shown very well in the canine use of this treatment, and we are in the process now of evaluating the specimens that we have from our trial to repeat and show that connecting the dots. The stimulation of the immune system in this particular way results in the improved survival against osteosarcoma, whether it's a canine patient or an adolescent patient.
All right, thank you. Taking the next question from Silver Turkan from Citizens JMP. Congratulations on the data update with strong OS data. Where are we with respect to the FDA regulatory sign-off? What is the timeline to the remaining Type C meeting focused on the endpoint with the FDA? And then separately, can you set the expectations for the prostate cancer data update? So with regards to the FDA questions, I'll turn that over to David Brindley to answer.
Thank you, Gerald, and I'll leave you the prostate one at the end. From the viewpoint of the timelines for the FDA Type C meeting, following FDA's invitation, the company made the application for the Type C meeting recently, and that proceeds on statutory timeline. So within approximately 21 days, we should have a confirmed date for that meeting. You'll appreciate that the government shutdown may delay things there slightly, but I would anticipate that meeting will occur before the end of the calendar year. And then with regards to the regulatory sign-off, I'm not quite necessarily sure, Sylvain, what you mean by sign-off, but if you're referring to a regulatory agreement on items that go into the BLA for market approval, as we alluded to, for CMC and non-clinical, there's generally agreement on most topics there, with a few minor follow-up items that are being submitted as IND amendments moving forwards. And then regarding the clinical item, particularly around biomarkers, and then any confirmatory post-market study that's required, that's really the key thing which we plan to discuss with the FDA in this forthcoming Type C meeting. So, Gerald, I think that addresses the question, and over to you for the second part of Sylvain's question.
And I will turn it over to Robert with regards to the prostate cancer. Can you set the expectation for the prostate cancer data update? What data will be available from that, Robert?
So the prostate cancer program is just really in its nascency. It's just beginning. This is based off of an adaptation that of this Listeria vector that was specifically tailored for prostate cancer. I think that initial work has been done with this at an investigator-sponsored trial, and the data looks promising. We're going to do some additional data analysis around some of the immunologic data that was collected there. And I think that that's sort of the pilot study of prostate cancer. And then based on what we see there and determination of the strategy and meeting with prostate cancer experts, we will devise a development program that will find out, that will really inform us about the best way forward, the best path forward to develop this in prostate cancer. Similarly, some of the early data in prostate cancer in an early version of the prostate cancer construct in combination with Keytruda was leading to some very impressively prolonged survival in late-stage prostate cancer patients in combination with Keytruda. So we'll be taking a look at various aspects of prostate cancer disease and finding out where this new therapy may be able to fit in.
Hey, Robert, in that same light, can you expand on where you think we can go once we get approval, if we get approval for osteosarcoma with OST/HER2 into some of the other solid tumors?
Absolutely. So the idea here is wherever there's tumors that overexpress HER2, like the majority of metastases in osteosarcoma, this treatment can increase the immune responses, the immune response fighting against the progression of that tumor. In something like HER2 positive breast cancer, the best idea would be to go very early in the disease where the patients are healthy and maybe receiving trastuzumab or Herceptin or HER2 targeted therapy. The problem with those HER2 targeted therapies is they only work for a short time, typically, and then the patients they become resistant, and then the patients move on to the next one and on to the next one. Some of the preclinical work I'd done previously really showed that if you can combine this Listeria construct with trastuzumab or HER2 targeted agents, they work together synergistically, and the HER2 helps to knock down the tumor initially, and the Listeria vector helps to generate the immune system to find these cells that will become resistant and help eliminate them. And so how that would read out in the patient would be potentially prolonging the amount of time that Herceptin or the original HER2 treatment would be viable in that patient, and preventing their need to go on to the next treatment, which ultimately prolongs their life. And so the idea would be to combine with active agents early in disease and prevent the development of resistance, or perhaps prevent the development of metastases like brain metastasis, which can be so devastating for women with breast cancer.
Thank you, Robert. Taking a question from Kumar Raja from Brookline. How similar are regulatory packages for UK and USA? Can you please talk about timeline once the applications are submitted? I'll turn that over to David.
Sure, very happy to. So first part of the question, how similar the packages for UK and USA, and I'll throw in Europe to that as well, so you get an answer for all three parts. Fortunately, the majority of the packages are standardized per ICH. So there's a tremendous amount of transferability. So it's not like the company has to do 3 times the work. There are some different pre and post submission requirements, but in general, they're very, very similar. And that has 2 benefits. One is cost and time for the company, but the second then is as the various regulators come back with any questions, when we address those, they can be shared with all 3 regulators in the same format without any additional work. In terms of timeline, so that has 2 dimensions. The first is when we submit, and the second is when we get the outcome. I believe that Gerald has addressed the when we submit, so I'll focus on when we get the outcome. So from let's start with the US first. From a US BLA perspective, a standard review is about 10 months. The company is eligible for priority review by virtue of it being a rare disease and has fast track status. So that looks around 6 months or less. It has been possible to get conditional BLAs in 3 to 4 months historically for very rare diseases. Again, the shutdown is a challenge here, but the statutory timeline for priority review is 6 months in the US. From an EMA perspective, there are 2 types of submission to EMA. There's member state by member state, so European country by European country, or the centralized procedure where you go to EMA, do 1 submission and approval in all the territories. As a rare disease, you have to go through the centralized procedure, and the process for that actually started, I'm pleased to say, last week for OS Therapies. And the timeline there is that you submit to EMA, you get questions after 120 days, and the entire process is meant to be wrapped up in no more than 210 days, so 6-ish and a bit months. And then the UK follows the same process, except you get questions at day 80 and day 150, but all completed by day 210 as well. So they are the responses for how long it takes once they are submitted.
Thank you. All right, we have a question from Chad Messer at Lake Street Capital. What did you learn from the OSI FDA workshop on Friday? And what did the FDA have any particular focus on the issues? I know that there's probably multiple answers for this. First, I'll start with David, and then we can go to Robert and Paul.
Sure. I'm actually going to pass this to Andrew, if I'm honest, from our perspective, because I think it's important to hear from somebody who has recent experience as a regulator as to what was really said and what it means. So, Andrew, if I may hand to you.
I think the most striking observation again was that there have been a number of studies, particularly at earlier time points in the disease with osteosarcoma, but to date, there has been no progress. So it's not really from a lack of effort or endeavor, but I think prior to the study we're describing to you now, there hasn't been the right mechanism, the right key for the lock, as it were, to discover. And I think the second aspect is that we are being encouraged to clarify the data that we are using for overall survival because of the difference between single-arm trials and randomized control trials. So that's one of the things we've been working on very hard recently, so that we can then present the wealth of data that does actually exist on the overall survival in this condition.
I'll turn it over to Robert now. I know you also had some interesting findings.
Yeah, thanks, Gerald. It was really an important meeting to get all of the parties together, industry, regulators, and the academicians, really to talk about the problems associated with developing drugs in osteosarcoma. And I think probably the biggest take-home message is that the academicians have been using signal finding protocols the same design of a signal finding protocol for decades. And this design was developed during the time of chemotherapy and targeted agents, which both of those tend to act really fast to knock down a percentage of tumor cells, but don't necessarily translate into prolonged survival. Sometimes they do, sometimes they don't. What we really were discussing here, this sort of, now things have changed in oncology. Immunotherapy is becoming where we're starting to be able to see real differences. And they set the bar very high, admittedly so, in this signal finding trial to find active agents. And so when they use something like event-free survival as their primary screening endpoint, then in a drug treatment that maybe isn't as strong up front, but is stronger in improving overall survival, that model fails because it doesn't capture the real potential benefit until you wait a couple of years to see, overall survival. And so that's, I was part of the development of the first sort of immunotherapy, the first checkpoint inhibitor at Bristol Myers Squibb. And that was exactly what we saw there. It wasn't really a large initial response rate, but we saw this tail of the curve where these patients were surviving tremendously long. And that's exactly what we're seeing here in osteosarcoma. And we really discussed, in the room, whether or not this model is too stringent for certain mechanisms of drug. And they might be actually setting the bar so high that they're missing active agents in this disease. And so that really was something we discussed as a group with the regulators and the academicians and industry there. And I think that what we're hoping to do is promote a different screening protocol for future development in osteosarcoma that will capture agents like this one, which improve overall survival, but don't necessarily have quite as big of an impact, although there was a good impact in event-free survival.
Thank you. I think we'll go to Paul more to speak about the patient questions, because obviously there were a lot of
Yeah, one of the
patients
most important stakeholder at the meeting last Friday with the FDA and OSI was the patients. And I think you can talk about study designs and the failures and the efforts that have been made over the years, but in the end, parents and patients want to have live to fight another day, right? And so overall survival is the key component here. The FDA has been incredibly flexible with other rare diseases, with multiple single-arm phase 1, phase 2 trials, getting conditional approval over the last couple of years. We just want to make sure that the FDA is looking at osteosarcoma in the same way, and I think that was the consistency throughout the entire room and the entire conference. And with the overall survival data that we have, and the biomarkers, and Dr. Sweet Cordero from UCSF talked about the importance of the canine studies and the biomarkers associated with canines versus humans. As Robert likes to say, the genetic makeup of osteosarcoma in humans and in dogs is 99% similar or identical. And so if the biomarkers in dogs can help, just as we've always talked about, if dogs can help lead us in this disease, that'll be a fantastic opportunity for our patients.
All right, thank you. We did get one question from Jordan Willis. Are you currently in partnership or acquisition discussions? And if so, are potential partners waiting for specific regulatory milestones? Obviously, we can't get into non-public information, but Paul, I'll let you take that one.
Yeah, we've had extensive conversations with commercial partners. Clearly, us de-risking this for them is their interest, and that's what we're doing. We've had conversations around global commercialization, ex-US, specifically Central Europe. We've not had a lot of conversations around Southeast Asia, and we're hoping to do that over the next couple months. Because again, the incidence of osteosarcoma is consistent no matter where you are in the world. And there aren't any really good options for this disease. So a lot of opportunities. We've also been in very active conversations around the commercialization of the canine indication, which we really want to have out on the market, if nothing else, for proof of concept for the regulators for human osteosarcoma.
Right, Harrison, I think that's the end of the questions, so I'll turn it over to you to help us wrap up.
Thank you. Well, if there's no more additional questions, we'll go ahead and conclude this conference call. There's going to be a recording available on the website on the investor relations page in about 4 days, 4 business days. So thank you all for your time today, and we'll be in touch.